Have you ever wondered how something invisible to the naked eye can impact life on Earth so profoundly? Well, we're about to dive into the intriguing world of bacteria that does just that. Bacteria, these microscopic organisms invisible to our eyes, are omnipresent. They exist in the air we breathe, the soil we walk on, the food we eat, and yes, even within our bodies. They are so tiny that millions can fit onto the tip of a needle. But don't be fooled by their size. These tiny creatures are mighty. In the grand tapestry of life, bacteria are the threads that hold everything together. They play a vital role in various ecosystems, helping to decompose organic matter, recycle nutrients, and even regulate climate. In the soil, they help break down organic material into nutrients that plants can use, effectively making them the world's smallest farmers. And what about human health? Bacteria are not just the villains causing diseases as we often perceive them. In fact, many of them are the unsung heroes within our bodies. They help digest our food, protect us from harmful invaders, and even contribute to our mental health. There's an entire universe of bacteria living in and on us, which scientists call our microbiome. However, just like any other living organism, bacteria have their dark side too. Some can cause infections and diseases, and when they get resistant to antibiotics, things can get really tricky. But that's a story for another day. For now, let's marvel at the wonder of these microscopic beings. They've been around for billions of years, long before us, and will likely be here long after we're gone. They've survived in the harshest of environments, from the icy realms of Antarctica to the boiling hydrothermal vents in the ocean depths. So, while bacteria are minuscule, they play a gigantic role in our world. They're the unseen workforce keeping life ticking, and our understanding of them is key to unlocking many of the mysteries of life itself. So buckle up as we journey further into the fascinating world of bacteria in the upcoming scenes. Did you know that bacteria are as diverse as the stars in the sky? Just as the night sky is adorned with stars of different sizes and shapes, the microscopic world of bacteria is brimming with a variety of forms, each as fascinating as the last. Let's delve into this invisible cosmos and explore the major types of bacteria. You see, bacteria are primarily categorized based on their shapes and groupings. The three primary shapes are Cocci, Bacilli, and Spirilla. Cocci are spherical bacteria. Picture tiny pearls floating around. That's how Cocci would look like under a microscope. They can exist as single entities or in pairs, chains, or clusters, akin to constellations in our night sky. Staphylococci, for instance, are cocci that form clusters, whereas streptococci form chains. Next up, we have bacilli, or rod-shaped bacteria. Imagine a bunch of tiny, elongated capsules. They may exist singularly, in pairs or chains. Bacilli are the daredevils of the bacterial world, with some types being able to form endospores, tough protective structures that enable them to survive extreme conditions. Finally, Spirilla. These bacteria spiral like little corkscrews. They're the free spirits of the bacterial world, moving around in a unique corkscrew-like motion. Each bacterial shape has its own unique characteristics. For example, cocci are typically non-motile due to their lack of flagella, a whip-like structure that allows bacteria to move. On the other hand, most bacilli and spirilla are motile, with flagella enabling them to navigate their environment. Moreover, these diverse shapes and groupings are not just for show. They play a crucial role in how bacteria interact with their environment, how they reproduce, and how they cause disease. For instance, the chain formation in streptococci allows them to spread rapidly in tissues, causing diseases like strep throat. Just as we humans differ from one another, so do bacteria, each type with its unique traits. Bacteria truly are a testament to the beauty of diversity in nature, each shape and grouping bringing something unique to the table in the grand scheme of microbial life. Ever wondered how fast bacteria can multiply? Well, let's dive into the fascinating world of bacterial growth. The growth of bacteria is typically divided into 4 distinct phases: the lag phase, the log phase, the stationary phase, and the death phase. In the lag phase, the bacteria are just getting accustomed to their surroundings, not yet multiplying but gearing up for the big growth spurt. Then comes the log phase, where the bacteria multiply at an astonishing rate. This is the bacteria's prime time, where they're full of life and energy. But even bacteria can't keep up this pace forever. In the stationary phase, the growth rate slows down as resources become scarce and waste products build up. Finally, in the death phase, the bacteria start to die off, unable to sustain themselves in their current environment. And here's a fascinating note. Factors like temperature and nutrients can significantly affect these phases and the overall growth of bacteria. Bacteria grow and multiply at an astonishing rate given the right conditions. How do scientists manage to study these invisible life forms in detail, you might ask? Well, it's a fascinating process that combines art and science, known as the isolation and culturing of bacteria. Let's start with isolation. This is the process where a single type of bacteria is separated from a mixed culture. Imagine you're at a party and you want to have a conversation with just one person. You would lead them away from the crowd, right? That's essentially what scientists do with bacteria. They use a technique called streaking, carefully spreading the bacteria over a surface until individual cells are separated from each other. Now, once the bacteria are isolated, the next step is to provide them with a comfortable environment to grow and multiply. This is where culture media come into play. Just like you might need a cup of coffee and a comfortable chair to get your work done, bacteria need specific nutrients and conditions to thrive. There are different types of culture media, each tailored to the needs of different bacteria. Some are like a buffet, offering a wide range of nutrients for bacteria to feast on. These are called complex media. Then, there are selective media, which are more like a specialized diet designed to encourage the growth of one type of bacteria while inhibiting others. And finally, we have differential media that allow us to distinguish one bacterium from another based on their growth and color changes. Imagine being able to create a perfect world for each bacterium where it can grow and multiply, revealing its characteristics and behaviors. It's like an intimate interview with each tiny life form. This process, my friends, is crucial. It's not just about studying these microscopic beings for the sake of knowledge. The information we gain from these cultures can help us understand how bacteria cause disease, how they respond to antibiotics, and even how they can be used in industry or agriculture. Culturing bacteria allows scientists to study them in detail, leading to many breakthroughs in medicine and technology. So, the next time you think about bacteria, remember, there's a whole world of microscopic drama and discovery happening right under our noses. How do we identify different types of bacteria under a microscope? The answer lies in a nifty process called staining. Staining is a technique used in microscopy to enhance contrast in the microscopic image. It's like giving bacteria a color code, making it easier for scientists to identify and study them. Among the various types of staining methods, Gram staining holds a special place. Developed by Hans Christian Gram in the 19th century, this procedure is widely applied and remains one of the most valuable tools in the world of microbiology. So, how does this Gram staining work? Well, it's a step-by-step process. First, a heat-fixed smear of bacterial culture is prepared. Then it's stained with a purple crystal violet dye. After a rinse, iodine solution is applied, which acts as a mordant, helping the dye bind more firmly. Next comes the intriguing part. The slide is washed with an alcohol or acetone solution. This is the crucial decolorization step. Depending on the bacteria's cell wall structure, it will either retain the purple stain or lose it. Bacteria that keep the purple color are known as gram-positive. These have a thick layer of peptidoglycan in their cell walls which traps the dye. On the other hand, bacteria that lose the dye and turn red or pink when counterstained with safranine are termed gram-negative. Their cell walls are thinner and more complex, with an outer membrane that the purple dye cannot penetrate. This difference in staining is not just about colors, it's a reflection of fundamental differences in the bacteria's cell wall structure. This information is incredibly useful, not just for identification, but also for determining the bacteria's potential pathogenicity and the type of antibiotics that might be effective against it. So, the next time you see a colorful slide under a microscope, remember, it's not just a pretty picture, it's a window into the fascinating world of bacteria, brought to life by the simple yet ingenious process of staining. Staining is like giving bacteria a color code, making it easier for scientists to identify and study them. So, what have we learned about these microscopic marvels? We began our journey by defining bacteria, these tiny single-celled organisms that exist in their billions, forming a significant part of our planet's life. We explored the diverse types of bacteria, each with their unique characteristics and roles in the ecosystem. We then delved into the fascinating world of bacterial growth, understanding how these organisms multiply rapidly under the right conditions. We discovered the importance of culture media, the nutrient-rich environments that allow bacteria to thrive and grow. Our exploration led us to the isolation of bacteria, a crucial step in studying these organisms in detail. We also touched on the staining method, a technique that enables us to identify bacteria by highlighting their physical features. And so, we conclude our adventure into the world of bacteria, having gained a deeper appreciation for these unseen life forms. Remember, while bacteria may be invisible to the naked eye, their impact on our world is anything but small.